Good morning, everybody. My name is Sophie and I am a first-year genetics PhD student. This morning I'm going in to check on my cells. I want to grow up lots of them before I start gene editing. If they've grown up well over the weekend, I'll be doing the gene editing tomorrow. If they've been a bit more slow, it probably won't be till Wednesday. I also need to get on top of my molecular cloning. I've gotten kind of stage 1 and stage 2 done in previous weeks, and now I need to do the final stage 3. It's been a bit more of a complicated molecular cloning strategy than normal because we had 2 plasmids. We wanted to take an insert from one and put it into a different backbone, so we had to create compatible sites for all of that to come together. All right, let's see how it goes.
So this morning, all I had to do in tissue culture was split down my cells. This is a pretty standard procedure. I do it a few times a week. The idea is if my cells overgrow, it's going to be really bad for them. So I want to split them into 2 different containers so that they've got plenty of space to keep growing. First thing I do is trypsinize the cells. Trypsin is an enzyme that cuts the proteins that hold cells together or hold cells down to the base of my plate. Trypsinisation takes about 5 minutes in an incubator, and then you can see I've got the second plate that I'll move half of these cells into. So now all of my cells are sitting up in the liquid in suspension rather than down on the plate. So I put a little bit of normal nutritious media in there to stop the trypsinisation process before it harms my cells, and use the same pipette to split half of that volume across to the new plate. Then I'm just adding some nutritious media back in so that the liquid fully covers the base, and mix them around a bit to make sure the cells are evenly spread across the base of the plate. They go back into the incubator and I'll check them out again tomorrow to make sure they're still growing well.
Long day in the lab today. I had my DNase extractions. They seem to go well. I had high concentrations come out of that. So that was DNA extractions from the cell lines that I had edited. So I wanted to see if I could see the editing in their sequence. So the extraction itself seemed to go well. Then you do a PCR to amplify the extracted DNA, and then you do a gel electrophoresis of that PCR product. And only one of the bands of 6 edited lines came up on the gel. So I didn't get anything sent away for sequencing today because I only want to send off the products that come up nicely on the gel with a band. So the PCR will run overnight tonight, and then we'll check those PCR products on a gel. I'm hoping to see 6 bands tomorrow, and if that's the case, I will send all 6 of those PCR products off for sequencing, which was today's plan, but is now tomorrow's plan. No other plans on the radar yet for tomorrow. I do have my 10-week report for my PhD coming up soon, so I need to start putting some more time into that. All I've got so far is maybe 800 words of dot points, and it needs to be 1,000 words of actual report. See you guys tomorrow. Good morning, everybody. Today's plan really depends on how the PCR ran last night, so the first thing I want to do in the morning is make the gel that I can verify that PCR with. So ideally it will show bands in each of my experimental lanes. If we do get those bands, I'll be able to send those samples off for sequencing. If not, I'll need to sit down and have a chat with my supervisor about what could be causing this. And then we'll probably have to try a few different things to see what did it, or repeat the experiment again. So if I get bands on my gel, I'm out of here in an hour and a half. If not, who knows? We'll see how it goes. Hello, everybody. I am on my way home to grab some lunch. My gel did not work out, just not seeing bands where we're expecting to, although this time we saw no bands where we were expecting to. So last time 1 band showed up at the expected size, this time none of them. So we have a new PCR block, which is the machinery that cycles through different temperatures required for PCR. So now we're thinking maybe we programmed that incorrectly and we're missing a crucial step. We did double check, but nothing obvious. So I've got a new PCR running and it's in the old block. So if this one works, we'll know we've done something wrong with the new machine. But no answers yet, and it's a 2-hour wait while it's in the PCR machine. So I'm gonna go home and have some lunch. Good afternoon, everybody. So like I said before, I did end up coming back in. It was just very quick. I don't have time to run another gel of this PCR product before our seminar this afternoon. So I've just got my PCR product nice and cold so that I can run the gel first thing tomorrow. It takes about an hour to run a gel, so that lines up pretty well with tomorrow. I'm planning to do another round of CRISPR editing, which takes about an hour 15. So my gel should run just fine while I'm doing that. So still no answers as to whether it was the new PCR block, but we'll find out tomorrow. See you then. Good morning, everybody. I was up late last night getting my 10-week report together. So it's a bit of an outline of what I plan to do, mostly in this first year, but also broadly across the project. I honestly found it really inspiring to take a step back, looking at the broader aims of this project and how many people it could help in the future. So I'm going to try and get in there today and do 2 lots of transfections if I can. You don't really want to double the number of tubes involved in the transfections, because the longer the cells are in the buffer that you need to use, the worse they do. Especially as I'm new to this and going slow, I don't want to add more complication into that. So I'll try and do 2 different rounds. See you in there.
Okay, let's do some transfections. I am cleaning out the tissue culture hood and grabbing everything I need. Those are my cells that I split down on Monday, and in the rack is my trypsin. First thing to do is remove all that old media and then add in the trypsin. So this is trypsinizing the cells exactly like we did on Monday. Grab everything else you need, including the warm media that's been sitting in a 37-degree water bath for 30 minutes. Add that in to stop the trypsinization process and move all of that liquid into a falcon tube so that you can measure the concentration. That's what that big clunky pipette does. I then have to do a bit of maths to work out exactly how much of those cells I need to go forward with. That's then going to go into the centrifuge to spin down so that all of my cells will split out from solution from the rest of the liquid that I don't need. In the meantime, I'm starting to get my RNP mix together. It's kind of a matrix that is going to deliver everything that I need into the cells. So we've got some R buffer, and then we've got the Cas9. Cas9 is the enzyme that actually does the CRISPRing. Because it's an enzyme, I'm really careful with it, so when I'm done with it, it goes straight back onto the ice. So then I add the unique gRNA to each of those tubes. So each one of those tubes represents a slightly different guide RNA that I am testing the efficiency of. And then I'm going to prepare my 6-well plate. This is the plate that my cells are going to go into once they've been edited. Okay, suction up the liquid off the top of that centrifuge cell tube and then resuspend it in R buffer. The R buffer is the buffer the cells really don't like to be in for too long, so now you are on the clock. Alright, I've got the neon machine in the background. That is what will deliver the electric shock that is going to get my RNP mixture into the cells. So then we get the neon pipette and suck up some of the cell gRNA Cas9 mixture, pop it into the machine, press go, and wait and watch carefully. I want to make sure it doesn't spark at this stage because that would indicate that the transfection hasn't been very efficient and probably it's fried my cells. I also do it twice per each lot of transfections just to make sure that if one doesn't work optimally, there's still a chance that the other one does.
Hello everybody. So I didn't get to do my 2 transfections today. I was worried I wouldn't have enough cells, so it's better to just keep them growing healthy so I can do it either tomorrow or maybe a week from now, who knows. But it was one of my best transfections to date. I only got 1 arcing event, which is fantastic. I'm stoked. Also, my gel product came up, so I could visualize my PCR product with the bands on the gel. So it looks like we're doing something wrong with moving the protocol over to the new machine. I looked at it again, I could not see any differences, but that is a problem for another day. So I sent those off for sequencing and we'll be able to see what they look like. So I'm heading home now to keep working on my 10-week report. Most importantly today, I need to finally put a massive figure together. My lab uses Adobe Illustrator and Adobe InDesign to put figures together and make them look neat and tidy. And I haven't used those before. So I've kind of been putting off getting this figure together because I've just never even opened these programs before, so I know it's going to take some time to learn. So I knew I had to allocate like a serious block of probably 2 hours just to get my head around the programs. I'm also not done writing. I got a second draft back from one of my supervisors this morning. So once I've got the figure together, I will send that off. Then I will work on the words and the content, and then I'm sure it will be perfect. That's not true. It's still going to go to my other supervisor. Wish me luck at my desk today. I'll see you tomorrow. Good morning, everybody. I am just on my way in for a very quick session in the lab today. All I've got to do is refresh the media on my transfections from yesterday, but I'm also going to take some time to discuss the figures in my 10-week report with my supervisor. Sometimes it's just easier to talk about design things in person. So I won't be in long today, just enough time to chat about my figures and how to present the data I've got so far in my 10-week report. And then I need to get home and actually do the work. See you in a bit. Hello everybody, I am on my way home and it has just started to rain, so that is wonderful. I got the media on my cells refreshed and they're looking pretty confluent. I think they'll be ready to split by tomorrow. I also had a really good chat with my supervisor about my figures. I think I was making them more complicated than they need to be. They were starting to look very unclear at the size they're going to have to be. I think we've got a pretty good plan together for some really good looking images. I am going to work on those figures this afternoon. That's my job for the rest of the day. I'll see you guys tomorrow. Good afternoon, everybody. As always, Friday was pretty simple. We had our lab meeting this morning. It ran for an hour and a half. I didn't have much to say. I've been working mostly on my report. I don't really have any new data. It's all been preparing for the next round of transfections, like getting these new cells started, just very routine stuff. I also ducked in to split down my cells this morning, pretty standard Friday activities. And now I'm just out for a walk before I need to sit down and work on my figure at my computer all day today. I think I said earlier this week that I had to set aside like 2 hours to learn Adobe InDesign and Illustrator. I knew I had to allocate like a serious block of probably 2 hours. And the longer I play with this software, the more I'm realizing what a ridiculous underestimation that was. I'm not finding them intuitive in any way, and especially for Illustrator, a lot of the tutorials I'm finding online are for artists who want to create, beautiful pictures, not import graphs and organize things on a page. So I'm still working on that. I'll probably be working on that all weekend, but I am actually pretty happy with my 10-week report other than this figure. I'm sure my supervisor will have more comments and it will go through a drafting process still. It's not perfect, but I'm pretty happy with it. If you've enjoyed this content, please like and subscribe. Let me know what you'd like to see more of. I'll probably show you that more. See you next week.